let's, here we go. We're going to go ahead and get started now. Oh, that's big. All three of us here in Sevierville and where everybody else is. Frankly, this would have been a great day to do the whole thing from virtual. However, since no one got around to closing the campus, we have to do it this way. You know, my wife was excited. Hey, guess what? Sevier County's closed. I don't have to go teach today. Great. So, not that I don't want to be here, but it would have been a great day to be home. So, okay, we are going to go ahead and wrap up cells, and we're going to have enough time, I think, to go ahead and start on chapter 24, metabolism. Don't worry if you don't have the PowerPoints for those printed out. We're not going to get very far, and most of what you're going to hear is an expanded version about the organic molecules, the carbohydrates, the lipids, and the proteins. So, but we'll see how far we get here. So, anybody have any questions on anything? Here we go. Okay, then I'm going to go ahead and get us into cells. Hopefully we'll wrap this up today. Now this is where we left off Tuesday. And we're talking about translation. And translation has to do with taking the instructions from the DNA and turning them into a protein. Because the DNA never leaves the nucleus. The DNA is the cookbook. The DNA are the instructions. And instructions for all sorts of things in a cookbook, just like the instructions for every type of cell and every protein that every cell needs to do its job. So the DNA will copy a gene onto a strand of RNA called messenger RNA. So we have, let me, so I can get this to, no, I'll just talk about it. So the DNA copies, makes a, DNA opens up and there's it's a double strand, and one side of the strand is the gene that we're looking at. The other side may be a gene too, but it's not the gene we want. So we open up the DNA, since it opened up the strand. One side is the gene that we want, and so we'll make a copy of that using messenger RNA. We'll make a single strand. It will be a complementary copy. The messenger RNA is always the complement to the DNA. Then the messenger RNA will proceed to leave the nucleus and go out into the rough endoplasmic reticulum. Once there, the ribosomal RNA will create ribosomes to form around the messenger RNA. Now, the messenger RNA is a complementary copy of the DNA, and that means that means that if the first base of the sequence in the DNA is a C, is a cytosine, then the complementary base in the messenger RNA is going to be G. It's going to be guanine, because remember, cytosine always pairs to guanine, and when we're dealing with RNA, adenine always pairs to uracil. Uracil pairs to adenine, and guanine pairs to cytosine. They're complementary. The messenger RNA is complementary to the original DNA strand. So we get the messenger RNA out in the rough endoplasmic reticulum, and we have the ribosome starting to form around it. But now we still have to get the amino acid there. And this is the role where we are in this slide. The transfer RNA is a structure made up of nucleotides, just like everything else, just like the messenger RNA and the ribosomal RNA is. And if we look at it, you know, I think I said this the other day, that this is probably the fanciest structure I've seen of a drawing of transfer RNA. Most textbooks just say it looks like a fish hook like that, but it's all made of nucleotides. These are all the bases along in here. And at the bottom of the curve, we have 3 bases, just 3, not anything else. This is called the anticodon. Now, the anticodon is the complement to the codon on the messenger RNA. Remember, here is messenger RNA, and we have clusters of 3 bases at a time. In the DNA, we call them triplets. Here, we call them codons. So if your codon in the messenger RNA is, for example, cytosine, 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 the anticodon on the transfer RNA, there's another one right there. The anticodon to this is going to be GGG. The anticodon is the complement of the codon. For every codon in the messenger RNA, for every codon will have an anticodon that is the complement of it. So if the codon's first base is A, then the anticodon, the first base will be U. If the codon second base is G, then the anticodon is going to be C. And if the third codon, the third base in the codon is U, the anticodon will be A. So it doesn't matter. Like, for example, the anticodon is UAU, the codon would be AUA. Since, you know, in RNA, U pairs to A and G pairs to C. Always. Those are the complementary pairings. So the anticodon is the complement of the codon. Now, attached to the transfer RNA, at the other end of the transfer RNA, let's get this, okay, we have the amino acid. Now, the amino acid pairs to the anticodon that's going to, and it's attached to that transfer RNA. It is going to be specific for that anticodon. So whatever the anticodon is, it's dragging a unique amino acid. Now, we saw on that previous chart, where are you? There you are. This chart here shows you that there are 64 possible combinations. Have you committed it to memory yet? That was a joke. So lame joke, but okay. So no one commits it to memory. When you have a very nice table like this, you don't need to commit it to memory. And I will, frankly, I have never, ever in 18 years of teaching here, had anybody ever come up to me and say, hey, what is the amino acid for CCG? I don't know, but I can look it up. So I seriously doubt in healthcare that someone will ask you that question too. So, because if you ever get involved in dealing with the amino acids and the bases and the sequences, you're going to focus on that particular issue, like hemoglobin, for example. Anyway, first letter, second letter, third letter, each one of these codons are going to code for a specific amino acid. And this is how the transfer RNA does. The amino acid that corresponds to the anticodon is going to be dragged to the messenger RNA. So we can put it together, so we can assemble the chain. Because remember, all the DNA is the the DNA strands, the genes are nothing more than the instructions for the proteins. The order of the triplets determines the order of the amino acids. That's all it is. While it's very, it is almost as simple as it is complex when you consider that you just have, here's 3 bases, here's 3 more bases, here's 3 more bases in the DNA, and it continues on down, and each one of those bases specifies an amino acid. And if you build a protein following that code of bases for specific amino acids, you get a protein that's supposed to do what it does. So now, okay. The translation process, a simple answer is protein synthesis, making proteins. That's the simple answer, and it works just fine. Translation is making a protein, converting the instructions on the messenger RNA to an actual protein. And this takes place inside the ribosome. So, and it's actually a pretty elegant little process that goes on here. 3 steps to translation, just like there were 3 steps in transcription. You have initiation, you have elongation, so you build the protein, and then you terminate, you have a stop code. And just like in transcription, you essentially have a start code and you have a stop code. Because when you're making your protein, you can't start with anything, suppose you accidentally copied a few extra bases from the gene directly above you or below you. You don't want to start too soon. You don't want to stop too late. So you have a start code and a stop code that delineates exactly where start and stop is. It's like a start sign and a stop sign. So initiation takes place when the messenger RNA goes out to the endoplasmic reticulum and a ribosome forms around the messenger RNA. Let me go back here and stop this. Oops, did that yesterday too. Okay, anytime 91. Okay, so what we see here at the bottom, here is your messenger RNA. This is what's known as the small subunit of the ribosome. There is always a segment on the messenger RNA that is the start codon or the initiator. And it's always going to be for the same amino acid. It's always going to be for an amino acid known as methionine. So every protein starts with methionine. So, and there is a start sequence on the messenger RNA that will tell the transfer RNA with its correct anticodon to come and land there. And that's the first one. It's always the first one. I don't know why it is, but it's always the first one. And everybody does it the same way. Every living organism does this exactly the same way. So here the anticodon is UAC. The codon for that would be AUG. And if we look right there, there is the codon. So we're always going to have that first codon of AUG on every messenger RNA because the first amino acid that's brought in is always going to be methionine. Now, as this is happening, a large subunit will come down on top of this. This is the large subunit. Actually looks like a big hamburger bun. And it surrounds the messenger RNA and it has sites for the transfer RNA to land. Let me clear this off. Back this up, there we go. Now, so here's our messenger RNA, there's our start codon, there's our small subunit. Here comes the transfer RNA containing methionine. And that's the large ribosomal subunit, the large subunit that lands on top of it. You'll see that this first transfer RNA contains methionine. The anticodon binds briefly to the codon, because they're complementary. So they're going to bind together just like the DNA strands do. So they bind together, and now we've got a methionine there. So what will happen next is a second transfer RNA is going to come down here. Here comes the second transfer RNA with amino acid number 2, and its anticodon is going to be CAU. I got that right, because the codon is GUA. The anticodon to the complementary codon to G is going to be C. The complement to U is going to be A, and the complement of A is going to be U. So the second one will come in and land on that spot. And when the second amino acid lands there, the first amino acid will become tied to it. Let me show you in this next slide what's going to happen. Okay, so here's AUG. There's the very first, there's our start codon. There comes the small subunit. Here comes the first transfer RNA with methionine landing right there. Now a second transfer RNA will come in, and its anticodon is the complement to the codon. And the first amino acid and the second amino acid will join together with a peptide bond, and then we will kick out the first transfer RNA. At that point, the ribosome slides down the messenger, the mRNA, exposing another binding site. Let me go back and show you that again. Okay, there's our start codon right there. Small subunit, transfer RNA with its anticodon landing on the codon. Here comes the second transfer RNA. It lands on the second codon. When that happens, the two amino acids link together with a peptide bond, and that first transfer RNA is kicked out. Now the ribosome slides down, exposing a new binding site. Here comes the third transfer RNA, and it binds. And these two connect to the third one. And this process is going to continue all the way down the messenger RNA until we hit the stop codon. So once the ribosome forms, it just sort of chugs its way down across the messenger RNA, and because it has to open up. See, right now we've got two binding sites both filled. As soon as the first transfer RNA releases its amino acid, it's kicked out, and it'll go back and pick up methionine again. And then the second one is now, it slides over, it gives up its amino acid. The second one's kicked out, it goes and picks up, in this case, arginine. And then as a fourth one comes in, the lysine one will be kicked out, and the strand's going to get longer and longer and longer as we assemble this. So this elongation process, the ribosome just continues to slide down across the messenger RNA. We're going to keep doing this. And the reason it slides down is so that the old transfer RNA can now be kicked out and a new one can come in. And we continue this process until we get to the stop codon. This is elongation. Now it can be hundreds and hundreds of amino acids in length. For example, hemoglobin is 360 amino acids long. You can slide down there and it's perfect. That's great. Or it can be 50 amino acids in length. It's whatever the protein, whatever the size of the protein is. But eventually we will hit the stop codon, and there are 3 stop codons. Excuse me. The stop codons are UGA, UAA, and UAG. Don't know why they're all based on uracil, but they are. The easiest one to remember is UGA if you think of UGA. from the University of Georgia. We got an A&P faculty in Tazewell that would describe that the only stop codon worth knowing is UGA. He's a Georgia grad, so we tend to overlook that. But the point, though, is it's easy to remember. If you can remember UGA, then you know the other ones that are all U-based. There's only 3 of them. When you hit the stop codon, you stop. What will happen is that instead of bringing a transfer RNA down, let me show you what happens again here. Oops. Okay, let's hit the stop codon is UGA, and instead of a transfer RNA that comes in, you get a releasing factor protein that lands on that spot, and that detaches the entire protein chain that's been made from the messenger RNA. The protein's done. Now, it'll have to be processed a little bit before it can be used, but it's essentially finished. And right now, it is in its primary structure. Remember, proteins have primary, secondary, tertiary, and quaternary structures. Right now, it's a long chain of amino acids. It hasn't been turned into a 3-dimensional shape yet, but it's finished, essentially. And as soon as that releasing factor lands on UGA, then everything stops. And the messenger RNA breaks apart into its appropriate nucleotides. The ribosome separates. The transfer RNA go back and continue to reload with their appropriate amino acids, and they wait. They wait for another opportunity, you know. And since this is happening in thousandths of a second all the time, you know, these transfer RNAs are really busy going to every ribosome because they're all, you know, it doesn't matter what the anticodon is. The anticodon will always have that amino acid. And so you could be going to ribosome 1 and ribosome 300 and ribosome 5,000 and ribosome 1 million, still bringing the same amino acid to that spot in the order that you're dictated to. You know? So we recycle just about everything. So once we get to the protein and we release it from the ribosome, then it's pinched off in a vesicle and is taken to the Golgi apparatus, where it gets put together in other vesicles of the same kind of protein. You know, that's what the Golgi does. It sorts it all out, and then it can get released. And once it gets released, it may be, you know, shaped into its 3-dimensional structure or whatever it's going to do. And we're done. We've made our protein. Okay. The reason that we do it this way, I don't know why we do it this way, but it works. But the reason we do it this way is so that we take a strand of DNA. We'll take a strand of DNA with a sequence on it of codons. Here is a strand of DNA that has been opened up. And let's just say that we have a codon, we have a triplet DNA. We have a triplet on the DNA. I want to keep it real simple. And we're going to copy this triplet onto messenger RNA. So what's the complement to adenine in RNA? It's going to be uracil. The complement to cytosine is guanine. And the complement to guanine is cytosine. So there's our messenger RNA. Now the anticodon, the transfer RNA anticodon is going to be A, C, G, G, G. The transfer RNA anticodon is the exact sequence of the original DNA. So we go through this complementary, we make a complementary copy of the DNA, and then we make a complementary copy of the complement. The DNA to the RNA, RNA to the transfer RNA, because the anti-codon sequence is exactly the same as the DNA sequence. So we're following the sequence specified in the DNA, but we have to do this double copying to get to that. We have to make everything complementary. And so our anti-codon is identical to the DNA. If the DNA would be cytosine, 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 the anti-codon for that is going to be cytosine, cytosine, cytosine. Because we've already gone through, DNA to messenger RNA, which is complementary, and then we take the complement of the messenger RNA on the anticodon, and we're back to the original sequence. So the DNA specifies every protein. Now, problems that occur. Mutations, there's different things that can cause mutations. A very common mutation is caused by ultraviolet radiation. You cause a substitution to go into the DNA. You get exposed to too much sunlight. Maybe you're exposed to ionizing radiation accidentally, or you're exposed to an industrial chemical, or any of those sources. Perhaps exposed to a virus that's a cancer-causing virus. Any of these sources. And a very simple mistake occurs sometimes when you're exposed. It doesn't mean you can't breathe the air or drink the water. Bless you. It just means that occasionally you may be exposed to something that might have an impact. But a substitution pulls out the correct nucleotide base and puts in a different one. So for example, the original strand up here at the top, on the first triplet, the original strand is cytosine, guanine, adenine, CGA. But if we substitute in a different base, now we get, instead of cytosine, guanine, adenine, we get cytosine, cytosine, adenine. Now, is this a significant problem? Well, it depends. It depends on what it's coding for. If we go back and look at our chart, if we look at CCA, what was it? CGA, should have written it down. CGA and CCA. Okay, CGA, the original one, CGA codes for arginine. Okay, so CGA codes for arginine, but CCA codes for proline. We've now put the instructions for the wrong amino acid in that sequence. Now, sometimes it doesn't matter. If you had CGA as your original triplet base, and you substituted in, instead of CGA, you substituted in CGC or CGU or CGG, it would all be the same thing. We have this redundancy built in, so even if we make a mistake in one of the bases, we'll still get the same amino acid, and it won't matter. But what we've done here with this substitution error is we've changed the whole sequence. We put in leucine instead of leucine instead of arginine, simply by one little mistake. Now, what will usually happen is if we make a mistake like that, the DNA strand, the enzymes that circulate up and down the DNA strands will slice out that mistake and replace it with the correct base. So the substitution mistakes are not that big a deal because they're usually fixed pretty well. And if it does occur, it usually only, it's usually going to occur at one location. So, if you have one little mistake at one location, then it's usually fixed. I mean, the error rate in a DNA strand after it's been processed to clean up any errors, the error rate for errors, okay, is about 1 in a billion. So it's a very, very small error rate. So maybe your DNA strand isn't going to work the way it is now, even if it slips through the repair. So that protein that would be made at that location isn't going to work. But we also are redundant in how many genes we have in our DNA. We have multiple copies of all of our genes. We're not basing it. We've only got one spot for hair color, and when that goes, we're out of luck. There's only one gene. No, we have many copies of the genes for every protein that we need. We don't just depend on one location. We can have an error occur at one location, and that protein doesn't get made because we're not going to make junk protein. If it's not what we want, we won't make it. But it can happen. These are not real. They may not be real significant mutations. On the other hand, here's one that is significant. This is hemoglobin becoming sickle cell hemoglobin. Hemoglobin is 360 amino acids in length, and at the number 6 spot on the normal hemoglobin, we have glutamic acid, and we can see the sequence up here. Look at the number 6 spot on the one marked normal. We have cytosine, thymine, cytosine pairing to guanine, adenine, guanine. But down below at the mutation, and this, for the mutation to occur, it's inherited. This is not something anybody acquires. It's an inherited disorder. A person is a carrier of sickle cell disease if they have the gene that can, if they have inherited one of the genes. You have to have both parts of the gene to have full-blown, to exhibit the disease. You can be a carrier of the disease if you only have one of the 2 genes. So you have to be, as they say, homozygous recessive. The disease is a recessive trait. You have to have both recessive traits. However, if you have it, then you have substituted valine in for glutamic acid at the number 6 spot. A little mistake made very early on in the sequence, and yet it destroys the carrying capacity of oxygen by in red cell in hemoglobin by about 80%. It causes the hemoglobin to become twisted around and brittle in its structure in a crystalline sickle shape, which causes the red blood cell to take that shape. It makes it very painful to exercise and do any kind of activity because red cells are supposed to be squishy like big red water balloons. And they can squish through tiny little, the capillaries, our tiniest blood vessels are very small, and our red cells go through that and they squeeze through and pop out the other side, and they're fine, they're happy because they're very elastic. Unless they're sickle cell red cells. They're brittle and they're sharp and they're pointy and they hurt when they go through here. So your patient doesn't have good oxygen delivery. They don't fight infections well. Their joints always hurt. They don't travel well in pressurized aircraft, so they don't fly well. All sorts of things like this can occur. Shortened lifespan, that and the fact that they're a carrier and they can pass those genes on to someone. Now, if you look at the mistake here, valine is very, very similar to glutamic acid. Glutamic acid is GAG and valine is GUG. one little mistake. Let me go back to the big chart. Here is valine, GUU, GUC, GUA, and GUG, and glutamic acid over here is GAA and GAG. Two choices for glutamic acid. Valine has 4. They're very similar. And what happens though is we have one mistake in the original strand that gives us GUG. So we get valine in here. This is a substitution mutation that is significant. Many of them aren't, but this one is very significant. And it happens. It's a recessive disorder. You have to inherit both genes from your parents to express the disease. If you only inherit one gene, then you're a carrier. And you may or may not have any symptoms at all of sickle cell. Now, another problem, frame shift. Frame shift is very dangerous. A frame shift mutation occurs when we actually stick a whole new set of nucleotides in at a spot where we have our DNA strand at the top there broken up. We have it marked out in codon in its triplet codon. Generally, we generally use codons for both DNA and RNA. We can say triplets so we don't get the DNA and the RNA mixed up, but we generally end up saying codons. So we have our triplets on the top strand, and down at the bottom, the second strand, we see for some reason, after the second codon, a second triplet, we've inserted a nucleotide pair, guanine and cytosine. That changes the makeup of that particular triplet. The original triplet was, adenine, thymine, adenine, with its complement of thymine, adenine, thymine. Down below, now we have guanine, adenine, thymine, and cytosine, thymine, adenine. We've slid everything down one spot. So now the whole makeup of the bases is going to dictate a different amino acid. Instead of specifying AT, let's take a look. ATA in the top. ATA, adenine, uracil. Okay, it should be specifying for isoleucine here. Well, what we're getting, let me go back to the, see what the second one was. So this is isoleucine, but now we're getting GAT, which is giving us GAU. Aspartic acid. So we're putting in the wrong amino acid in here, and everything below that insertion is wrong. This one is no longer this identical to the top strand is now changed by putting in those bases. Everything slides down one. The triplet sequence is altered, and now we have a whole chain of nucleotide bases that are out of order. And are we going to make real protein out of it? Well, no, because the instructions are, we don't want that protein. We want, whatever, hair color or, an enzyme. All of a sudden, the gene is producing a protein that it doesn't want to make. The gene is essentially destroyed by this one mistake here. By putting in a new set of nucleotides, we've destroyed the gene. We don't make the protein anymore. Now, did it happen throughout, or was it just one location? We don't know. This may be a routine event over our lifetimes as genes wear out, because genes can probably wear out. But once we stick the nucleotides in there that don't belong there, and they stay there, then the whole gene is destroyed. Okay. Now, our cells depend on our DNA. That's obvious. Every cell, and I've said this several times, every cell has an exact copy of the DNA. Everybody's DNA and everybody in every cell in our bodies all has the same DNA in it, except red cells. And even then, we did have DNA, but we kicked it out when we kicked out the nucleus. So it doesn't matter if you have a liver cell or a skin cell, all the DNA is in both. It's just what's turned on and what's turned off. Again, go back to the cookbook analogy. The instructions in the cookbook, if you're busy making biscuits, you're not going to be paying attention on how to make mashed potatoes or something, ridiculous, whatever. If you're making Jell-O, you're not going to be paying attention on how to make a cake. Those instructions are somewhere else. They're there, but you're not going to use them. If all you're doing is making Jell-O, for whatever reason, maybe it's a church dinner, and you got to make lots of Jell-O salad. You're going to follow the Jell-O instructions. You're not going to be looking at the cake instructions or the, baked chicken instructions or whatever, but it's still there. Liver cells do liver cell things. Skin cells do skin cell things. All of our genes, all of our DNA is exactly the same in every cell. It's just what is turned on and what is turned off. When we were developing in utero, our cells were pre-directed as to what they were going to do. We call that they were differentiated into skin, brain, liver, heart, lung cells. Once they started to grow, then they grew into those various shapes. They were predetermined as to where these cells ended up in our embryonic state for the first 2 weeks of our life. Where those cells ended up in our embryonic state dictated what they were going to become. So they were already predetermined. If those cells were to move into another part of the embryo, then they'd become a different thing. So our cells are already, it's all specified what they're going to do very early on. In AP2, we're going to talk a lot about that. We are 7 days after fertilization. We are a structure of about 160 cells that is very organized already. The statement that we are just a blob of cells, an undifferentiated cell mass, that definition applies for about the first 3 days after fertilization. After that, we are very organized. Consider the fact that our heart starts beating 21 days after fertilization. We are still microscopic, and yet we have a beating heart. So just consider that. Okay, now cells will divide and replicate their DNA at different rates. Nerve cells don't replicate, muscle cells don't replicate, but skin cells do, and scar tissue does, and liver cells do, and skin cells do all the time. I think I probably repeated myself there. Every time we're injured, every time we cut ourselves, we repair the damaged tissue without having to think about it. There's nothing we do when you get a cut or a scratch. My dogs are always tearing up my hands, but I don't care because they're my dogs, and I know it's going to heal. I don't have to, other than stopping bleeding, I don't have to do anything. It's going to heal. All of our injuries are like that. Our bones will heal without us doing anything to them, even if we don't get them set right. We have what's known as hyperplasia. Cells will grow more when they're needed. Muscle cells will get bigger if we exercise. Bone cells will get bigger and more of them if we exercise. And if we don't, they're gonna shrink. And we're gonna, like some of the cells, like bone cells, will lose. Hyperplasia, anytime we have hyper, it usually means an increase. Hyperplasia is cells that increase in number or in size or both. Like bone cells will increase in number and get bigger. Muscle cells, we won't get any new muscle cells, but they'll get bigger when you exercise. If you don't exercise, we have atrophy. The cells will either break down and go away like in bone, or they'll get weaker like muscles. Astronauts in the space station always have a problem with their muscles shrinking over time because there's no gravity. And despite the fact that they can exercise on resistance equipment, they don't get that constant pull of gravity on our bodies. And so their muscles will weaken, their bones will shrink in size. And if they've been in space for a long time, like over a year, they may not be able to support their body for a few days once they're back on Earth. So because of the pull of gravity is more than their long bones can withstand. Now, what determines the age of a cell? And I mentioned this earlier when I talked about telomeres. There's 4 different theories here. Wear and tear on cells. What I'm talking about is a typical cell is capable of supporting 50 replications over the lifetime of what we call the cell line. So if you have the original cell, it will divide and give you 2 daughter cells, and then they'll divide and they'll divide and they'll divide, and it can last out for about 50 replications. And then it stops. Because remember, you're still using the original DNA is continued, in some form or another from the start to the finish. Because through semi-conservative replication, you're always using half the original DNA of the parent cell to go into the daughter cell. So everybody's still containing some of the original DNA. And the cells wear out. Maybe that's why the cell line stops. Or the mitochondria in the cells become useless, or the immune system attacks the cell. But my personal favorite are the telomeres. Telomeres are the ends of the DNA strands. So the ends of the chromosomes. Well, that's the ends of the DNA strands. I think so it was on the right spot here. Okay, here we go. This is a telomere. This is the end of a DNA strand. This is another end of a DNA strand here, and I probably drew it too high. There we go. Still can't see it. Move over here. There we go. DNA strands. This section here and here represent the end of the strand. These are telomeres. Telomeres are like the caps on our shoelaces. They protect the ends of the shoelaces so they don't fray. Every time a strand of DNA replicates, some of the telomeres are chopped away. And so as we get closer and closer to the end of the telomere, the DNA side of the telomere, as we get closer to this segment down here before the DNA strand, we have less and less of a lifetime. The telomeres are worn away, and as they wear away down to nothing, then the strand stops. So the telomere shortens each time there's a replication. That's our timer. How long are this cell line going to survive? And we don't grow telomeres back, except cancer cells. Cancer cells cheat. They grow back their telomeres. So they can always, they will continue to grow. There are cells from a cancer patient. She died of cervical cancer in the 1950s, and her cell line is still alive in various research institutions that are studying different types of cancers, because the telomeres never wear out on that. It's a very famous cell line that has been used millions of times around the world for research. So anyway, so that is I tend to go along with the view that it's the telomeres wearing out. So, okay. And that is the end of cells. Okay. So let's take a minute. I'm going to shift over into let me get into yeah, let's see if I can find the correct chapter. Now you probably are wondering why our chapter is called chapter 24, because that's the chapter in the book we used for years, and we just sort of stuck with the name. And believe it or not, in most A&P books, the chapter on metabolism is still somewhere right around the 20s. Now, why do we bring up chapter 24 here out of sequence from the cells? Well, the reason we do is because there's two reasons. One, this allows us to discuss those macromolecules like glucose again and fats and proteins, but it also explains how the cells release energy from glucose. The whole process of cellular respiration. And so I like to think we're getting rid of we got rid of the ugly chapter on chemistry. Now we're going to get rid of the ugly chapter on metabolism. So, and then we're done. We're done with ugly at this point when we finish this chapter, and it's short. And then we go into bones and muscles, which is a whole lot more interesting. But oh, did I say that? I'll have to edit that from the video. So, okay, let me bring this up now. Nutrition and metabolism. Why do we eat? To have energy. Yes, we eat to have energy. We like to eat. We all have our favorite foods. We all have our foods that are less than favorite. When I was a kid, I never liked any kind of beans. Now, I do like some types. I still can't eat. I still won't eat lima beans. Thank you. Yes, right there. But I do like navy beans and pinto beans. I like those. Don't like kidney beans. Can't stand kidney beans. So to the point when I make chili, I don't use kidney beans. I don't use any beans in my chili. It just gets in the way. Anyway, nutrients are what we eat. We get our energy sources in our food, and the nutrients that our bodies need are broken out into 6 different categories. We have the carbohydrates, the pastas, the starches, the potatoes, the apples, the biscuits, the things we like. Lipids, the fats. Fats are a greater energy source than carbohydrates are. And if it wasn't for fats, a lot of our food would be really bland and difficult to eat. And of course, the proteins. We need proteins for our muscles. Our muscle cells are made of protein. We need to have the ability to contract and move. We have to make enzymes to do chemical reactions in our bodies. These are all really important to us. We also need to have vitamins and minerals. Vitamins like vitamin D, vitamin C, vitamin E, vitamin A. These are all important vitamins to us that help the enzymes do their activities. Vitamin D, for example. Vitamin D is very important to us. Vitamin D allows us to absorb calcium in our small intestine. Without vitamin D, we can't do that. And we need calcium for our bones. We need calcium for our muscle contractions. Minerals, we need calcium. We need iron. We need iron for our hemoglobin. We need calcium and phosphorus for our bones and our teeth. We need salt. We need sodium and potassium for our fluids. So these are important vitamins and minerals. We know in other elements, but that we have to have in our diet. We also have to have water. Our bodies are 70% water. Most of our food is made of water. You know, if you like leafy greens, if you like spinach or romaine lettuce or kale or any of those kind of, like the salad, you know, all those are something else I didn't like when I was a kid. But they make, you know, great as salads or you know, like who likes spinach Maria? You ever try spinach Maria? You know that.
Spinach dip?
Yeah, it's it's Parmesan cheese and spinach. Yeah, oh yeah, and every restaurant has a variation on that. You know, sometimes it's a side dish, sometimes it's a dip. You know, who doesn't like that? You know, yet if you told me when I was 8 years old, you don't like it. Oh, you ought to try it sometime. Holston's has it too. You know, you ought to try it. Aubrey's, yeah. If you told me when I was 8 years old, I'd eat spinach and enjoy it. You know, no. So, or anything else. But most of those foods are filled with water. You know, apples and oranges and potatoes are all filled with water. And we need that water. That's a big portion of the water supply that our body requires. So this is the Department of Agriculture's food pyramid, supposed to help us understand what we're supposed to eat a lot of and what we're not supposed to eat a lot of. And we start out with the oils and the whole grain foods at the bottom, and we end up with the rice and white bread and potatoes and pasta at the top. With you know, where does, let's see where meat falls in. We got chicken and fish. You know, the red meat's up at the top too. And you know, there's something wrong with this chart, but you know, it's very hard to generalize eating habits for somebody. How can you look at this and really get an idea of what's enough and what's too much? You know, some people take the idea of, well, you know, all the evil stuff's at the top of the pyramid, we can't eat it ever, you know, or you can overdo the use of the oils and the whole wheats at the bottom of the pyramid. You know, it basically what you advise your patients is that they can pretty much eat whatever they want to, as long as it's not excessive. You know, as long as they aren't eating excessive amounts of it, and you'll know that, you know, by their blood work and their body shape and their body appearance. You know, whether they're, you know, indulging, and there's nothing wrong with indulging on something now and then. You know, if you like pizza, you know, and most people like pizza. You know, everybody has their favorite types of pizza. You know, and there's nothing wrong with sitting down and enjoying it, as long as you're not going to do it every night. You know, and even then, you know, think about Michael Phelps, the swimmer from the, you know, he won all those gold medals in swimming. He would consume 10,000 calories a day. You know, he would eat from McDonald's and other places, 10,000 calories a day, but he was burning off about 12,000. So he could eat whatever he wanted. You know, he doesn't, if he's not in training now, he's not going to be eating like that. You know, what is moderation? Well, it's, you know, enough to enjoy it, but not to overdo it. You know, there's and that's the hard thing too, is what is going to be moderation. So anyway, the Department of Agriculture has put together a pyramid every so often. The last version of it was an inverted pyramid, which made no sense whatsoever. So I always show this one. Now that you've seen it, you can forget about it. However, the point of eating is to gain energy. Whether we're taking in carbs or fats or proteins, we're going to convert them into glucose and then we're going to burn the glucose, and we're going to release energy in the form of ATP. That's what we do. And we measure the energy in food in calories. It's actually kilocalories. If you have, I have nothing up here with food with calories on it, but if you had, one day nobody's here, you know, but if you had a soft drink, if you had a Coke or a Pepsi or something, you would have something on the bottle that said like, you know, 220 calories. Or you had some sort of snack, tells you the calorie content in there. That is the number of calories. That is represents the number of amount of potential energy in that food item. 160 calories, 140 calories, 130 calories. An apple or a potato is about 60 calories. You know, a bag of fries, 300 calories. You know, whatever. It's how much energy, how much potential energy is available in that food item. Now, we're not going to get to use all that energy for us. We're going to lose part of it, but we can get what we need out of what we eat. Now, what is a calorie? It's defined as the amount of energy necessary to raise the temperature of 1 kilogram of water by 1 degree Celsius. And in food science, there are devices that are used to measure this, so that the calorie values that we get stamped on food labels are pretty accurate. They're pretty consistent. You know, it's a whole field of science on that. So I would think it would really probably ruin some people's desire to eat things, if they knew how many calories were in everything. So. Now our carbohydrates, we talked about this already. The carbohydrates are where we get a lot of our energy. Most of our carbs come from plants. The sugars, the monosaccharides, the disaccharides, we find in fruit, we find in cane sugar and beet sugar, as well as in milk and even in honey. Honey is a great source of sugar too. I think I probably made this point earlier. There are two different types of sugar sources of sugar: sugar cane and sugar beets. And the only way you can tell the difference is on the label where it says pure cane sugar, it came from sugar cane, and it says pure sugar, it came from beets. After that, it looks exactly the same and tastes exactly the same. It's the white sugar we're used to. Plants also provide a carbohydrate called cellulose. Cellulose is the covering of the kernel, for example. If you have wheat bran, if you have wheat, when wheat grows, it's found inside the part we eat is covered by a strong, tough material that protects the kernel. That is cellulose. We can't process cellulose. It's the stuff that forms the cell wall of the plants. You know, if you like corn on the cob, if you like any kind of corn, once you eat the corn, the yellow covering or the white outer covering of the corn kernel passes through your body. It's made of cellulose. The material on the inside of the kernel, we can process that, we can eat that, but the yellow covering of the kernel will end up in our fecal matter as it leaves our body. We don't process that because it's cellulose, and our bodies can't deal with cellulose. We call it fiber. It's the indigestible portion of bran flakes, you know, raisin bran muffins. Cheerios are made of oat bran. Now, that is the outer covering of the oat kernel. We can't process that fiber, that oat bran either, just like whole wheat, the whole wheat covering or the whole covering of the oat kernel. Can't process it. It functions as fiber, as what we call roughage. Now, wheat fiber has a very cleansing effect on our colon. It helps to clean it out. It helps to push fatty deposits and fecal matter through there because it's not digested and it comes through in a fairly large mass. Oat fiber, on the other hand, actually does what they say on TV. Oat fiber helps to increase the production of the high density lipoproteins, the proteins that carry cholesterol back to the liver for destruction. So when they say that Cheerios are a heart healthy cereal, well, so is any oats based cereal. It doesn't have to be Cheerios. It can be any Cheerios knockoff, you know. It can be Oreos or whatever, you know, that they call them it, you know. The stuff you go to Food City and to the bottom shelf, there's jumbo bags. You know, they look like Cheerios. They don't taste like Cheerios, but they have the same ingredients, you know. Oatmeal does the same thing, you know. Oatmeal raisin cookies don't classify as a health food, you know. And you think it's got oatmeal, it's got raisins, it's got to be good for you, you know. It's not. Well, it probably is. I don't know. So the bottom line, though, is that we do need to get carbohydrates so we can get glucose. The disaccharides, the monosaccharides, the polysaccharides in the form of starch, we can process that just fine. We eat an apple, we eat a pear, we eat a banana, we eat potatoes, we eat breads, we eat pasta. We break those carbohydrates down to glucose. And glucose is what our cells use to make ATP. We actually burn the glucose. We oxidize the glucose. And what we're doing when we oxidize glucose is we release the electrons. We release the covalent bond, the covalent shell electrons, the valence shell electrons. We release those and all their potential energy is available for us to use. We only get to use about 40% of it. That's still pretty good. So how many carbs a day do we have to take in? Well, we don't know. We don't know what the upper end is. We know what the lower end is. The lower end is about 100 grams of carbs. So that's like the minimum would be like 2 apples, you know, is about 100 grams of carbs. You can, you know, every food label will tell you how many grams of carbohydrates are in there. 100 grams is about 400 calories. You know, 1 gram of carbohydrates equates to 4 calories. So 100 grams gives you about 400 calories. Now, the average person should consume about 2,000 calories per day, because that's about what we burn. We burn about 2,000, so we should, you know, if we have 2,000 going out, we should have 2,000 coming in to maintain our weight, to maintain our functions. So if we eat 100 grams of carbs during the course of a day, we're going to get 400 of those 2,000 calories we need. Most dietitians would recommend somewhere in the neighborhood of, you know, about 150 grams, somewhere on either side of that, about 150 grams, but they should be complex carbs like whole wheats and, you know, the starches, you know, the pastas, not the simple sugars. You know, drinking a Coke to get your calories, the glucose you need to function, is not a good idea. Nothing wrong with having a Coke or any other drink like that. But if you're depending on the Coke for your source of your carbohydrates, that's not a good idea. Remember the commercial that the Betty White commercial, you know, you're not yourself if you don't eat a Snickers bar, and they had Betty White in there. And the Snickers bar, and there's nothing wrong with the Snickers bar, But it's full of fats and carbohydrates. It's got some protein. It's not a bad candy bar because you've got protein from the peanuts, you've got oils from the peanuts, you've got oils and carbohydrates in there. It is not a bad snack food, but it does represent what we call empty calories because it doesn't take much to process the simple sugars that are in the candy bar. And the faster we process a sugar, the less likely we are to get a lot of energy from it. We like to have the starches, the polysaccharides, so we can break them down slowly and slowly release their energy. Whereas a Snickers bar, you're going to get that rush of sugar real fast, and then you're going to crash. It's the same thing as having you're coming to school and you're hungry, so you stop and get a couple of donuts at Krispy Kreme or Dunkin' Donuts, and you have a cup of coffee, and you get your caffeine buzz, and you get your sugar rush. Because if you've ever seen how they make donuts, you ever see the sugar bath at Krispy Kreme, they have this huge conveyor belt, and they have thousands of donuts coming across the conveyor belt, and they've already been fried. I guess that's how they make donuts. They fry them in these big vats of fat, and then they come through this belt, and there is this bath of liquid sugar that comes down and covers them. That's why when you get them when they're still hot, and the red light's on, the sugar is just started to crystallize on the surface, and I mean it's delicious. Who hasn't been there, right? You know what I'm talking about, right? Yeah, and they're so soft and chewy and tasty. Like 1, 2, 3, and like, hey, they're all gone. Let's get some more. And it's just this liquid bath of sugar, and it's all simple sugars, and so you get this big rush of sugar and nothing behind it. There's nothing wrong with Krispy Kremes. I like Krispy Kremes. I don't go there very often, but I do like them, believe it or not. So, but those are the wrong kind of carbs. There's nothing wrong with having them, but but not as our sole source of carbohydrates. The carbohydrates we need are the complex carbs, the pastas, the potatoes, the starchy potatoes, the starchy fruits like apples and pears, but even bananas. We need the carbohydrates that show up in milk and in dairy product, the things that we can break down and release the energy slowly. Because if we release the energy the faster we release the energy, the less likely we're going to be able to use it for something. It's like when you are, if you're using a charcoal grill, okay, and you put your charcoals in there and you soak it with lighter fluid and you toss a match into it, and you get this huge jet of flame that, and you have to grow your eyebrows back, after that because it's so powerful. Are you going to cook on that? Or are you going to have to wait till the coals cook down and cook on the coals? Or you use a gas grill. Are you going to, are you going to have it full blast, with the flame shooting up through the grill? Or are you going to keep it down to around 300 degrees when you're cooking on a gas grill? We cook our foods slowly so we can, so they can cook better. If we burn our calories off real fast, we're not going to get to use them. In our cars, we don't release all the energy in our gas tank at one split second. That'd be really exciting, but it wouldn't be a lot of fun, when it was over. I guess it depends on your car too. So the faster we burn our calories, the less likely we can use them. So the simple sugars burn quickly. The complex sugars take more time. We get more time to use that energy to do things. So how much do we actually eat on a daily basis? Well, we're supposed to take in about 150 grams. We usually take in about 300 grams. So probably 1,200 calories a day from is in our carbs. So that's sort of the American standard, if you will, of what we take in. And let's see. As I already said, the complex carbs, potatoes, the cereals, the pasta, the nuts, the rice, starchy foods. You know, we try to stay away from, we try not to eat a lot of the highly refined stuff, the soft drinks, the candies, even fruit. While fruit is full of complex carbohydrates, it's also full of sugars, the simple sugars. That's what makes fruit taste really good. As the fruit ages, it tastes better. It tastes sweeter because the starches. Let's say you have an apple that has aged, and it's sort of funky looking there, and you're like, I don't want to eat this. If you were to bite into it, you would find it's very sweet. It's like the ongoing argument about bananas. Some people like their bananas green. Some people like them yellow, and some people insist the only way to eat a banana is when the skin is white. Okay, we have that discussion all the time. Jillian, our lab tech, is a big fan of brown bananas. Dr. Grice and I are in favor of green bananas. But the brown skinned banana is going to be much sweeter because the banana has had time for its sugars, its starches, to break down to simple sugars. And so it has a much sweeter flavor. There's a reason why we use the brown skinned bananas in baking, for banana bread and muffins and rolls and stuff like that, because they're sweeter. So we have to watch out. Just because it's a fruit doesn't necessarily mean it's good for us. Something else that always surprises me is immature vegetables, like the little tiny ears of corn that you get in Chinese food, or vegetables that they're not ripe yet, or they haven't reached their mature size. They're full of sugars too. Who knew? And we tend to call all this stuff that empty calories, because they don't. We burn them fast, and we don't get a lot. We don't get a lot of long-term energy from it. And the consequences of these empty calories are that if we release a lot of sugar at once that we can't all process, what's going to happen to that sugar? We may end up storing it in our adipose, where it becomes fat. I mean, we'll always store sugar as glycogen in our liver. But once we fill up the glycogen storage, then we're going to start storing it as fat. And that's the problem with eating a lot of high empty calorie content stuff. Like, there's nothing wrong with eating it. It's just how much of it do we eat? Okay, that got us to lipids, and I can stop here, and we will pick up with this on Tuesday and move on. So anybody have any questions? Okay, well then have a good weekend, and I will see everybody next week.